Hey guys, good morning. As you see, my child is making noise in the background, but you know, my husband will get him in just a moment. While I wait for some people to get on, go ahead and say hi. Hope everybody's doing good this morning. Shout out to two other test prep, AP prep type of accounts that you may want to follow as well. So we have the Absolute Recap. They're doing, good morning, Vicki. They do biology, they do AP Chem, they do US government, they do music theory, and they do physics. And so the Absolute Recap is actually a podcast, and every single podcast is less than 10 minutes. So if you're liking little podcasts, then they're actually your place to go. And the second person I want to give a shout out to is Marco Learning. Marco Learning, I did a quick look through. They do a lot of different subjects, but they do a lot of stuff with college prep. So ACT and SAT prep. They do AP Spanish Lang, they do AP US History, and I know they do AP Lang. They may do a couple other things, but I'm not 100% positive all of them off the top of my head. So, hi, good morning. I see I got about 6 of y'all. Today's goal is for us to talk about Unit 2. So Unit 2 has to do with the cell. Yeah, I agree, Vicki. Market learning is amazing. It's about the cell. And so it's about all of the different organelles that are within the cell and how they work together, and how they allow the different structures to happen, or the different functions to happen within the cells. And then also it has to do with membrane transport. So you should see a little question box in the bottom. So you can submit your questions down there if you have any questions that you want to ask. Before the session started, I asked anybody if they had any questions, and nobody had any questions at the time. Hey, look, the Absolute Recap showed up. Say, hi, Absolute Recap tour. So if you have questions, nobody submitted them ahead of time, so I'm kind of just going to free talk about organelles until you guys have questions. So real quick, we can kind of think about the different organelles that are within the cell. So the most important one, is the nucleus. So the nucleus is going to be more or less in the center of the cell. It kind of floats around a little bit, but it's overall held there by intermediate filaments that kind of keep it in place. And the whole job for it, hey, No World, is that it's going to be where we're going to see the DNA held. And so when we're talking about a nucleus, and we're talking about most of these organelles, they're all membrane-bound organelles, which means they're going to be in eukaryotic cells, because our prokaryotes don't have a membrane-bound organelle at all. And so if we see a nucleus, we immediately know it's eukaryotic. And so the DNA is held within the nucleus, which is this double membrane that surrounds it. There are these pores that are in it. And the pore is going to allow for the transport of various things. So for example, we can have enzymes moving in. We can have these things called transcription factors moving in for cell communication to say, hey, turn on a gene, turn off a gene. You're also going to find that the ribosomes, the rRNA and the proteins for the ribosome are all within the nucleus, and they're going to have to leave the nucleus in order to go through that. The proteins for the ribosome are not in the nucleus. It's just the rRNA, and it's going to leave to then form with the proteins to be able to make those ribosomes in the cytosol. So the big point about the nucleus is, one, it's going to store your genetic information. It stores the DNA. It's the site of transcription. Transcription is the process where we take our DNA and we are going to read the DNA to synthesize a copy of mRNA, or any other RNA, if you will. And so that RNA can then leave the nucleus to perform its actions. So another different organelle that we have would be the rough ER. So the rough ER stands for Endoplasmic Reticulum. It basically just like this mesh kind of network. The reason why they call it a reticulum is because back in the day, there was these purses that women carried that were called reticulums, and they were just basically a big net. And so when they were naming the endoplasmic reticulum, they said, hey, cool, it looks like that thing. Hey, Marco. And so they then said, hey, let's call it the endoplasmic reticulum because it's made of plasm inside of the cell. Hi. So the rough ER is going to have ribosomes attached to it. So because of the fact that you have a ribosome, which has a function of a protein synthesis attached to a membrane, it's going to allow the cell to make membrane-bound proteins. This means it's a protein that's actually in the membrane. It's also going to allow for you to make proteins that are inside of membranes. And so whenever we're going to secrete materials from our cell, they need to be within a membrane. And remember, if y'all have questions, go ahead and put them into the little question box. So we're going to see that the rough ER makes membrane-bound proteins, and then it also makes proteins for secretion. We also have the smooth ER. The smooth ER has a variety of functions. One of those functions is that it's going to allow us to detoxify. It's also going to allow for the storage of calcium, and it synthesizes lipids. And so you won't really see a lot of the calcium part until we talk about muscle contraction, because within a muscle cell, there's a certain smooth ER called the sarcoplasmic reticulum. And that actually is going to be where the calcium gets released from in order for us to be able to have the secondary messenger to allow the muscle to contract. The important part about the smooth ER being for detoxification is that in cells in which we're needing detoxification, so for example, your liver, you're going to find there's a large concentration of smooth ER. And the more that the cell has to use that smooth ER, there's going to be an increase in the amount of smooth ER. And so we would find that in individuals who have consumed higher quantities of toxins, that in their liver, they would find a higher concentration of smooth ER. And so the reason I'm going through this extension part is because the questions on the AP exam aren't going to be straight, what does the smooth ER do? It's going to give you a story. It's going to give you background. It's going to give you information about it. And you're going to have to apply it. So they may tell you that there's in the liver cell in an individual who has overconsumed toxins, what organelle would you find an excessive amount of? And you'd have to think, okay, well, in the liver, we're going to see detoxification. It said that we have a higher concentration of this certain organelle. Well, that must be the smooth ER because its job is to detoxify. So you kind of have to think through and work through that. So I'm going to pause on my little review because somebody has asked the question. Let's see what the question is. New follower here. Do I have a YouTube channel? No, I don't have a YouTube channel. It's just I do my review through my Instagram stories. So I like to just do every weekend I'll post review questions that I'm giving to my own students on my main account. And then in February, I start a big review session in which every single day I post review questions, and they'll be interactive, so you can actually click the multiple choice question on the story and all that. And as we get even closer, I do live stories. And so this year, I decided that I was going to do lives after each of my units that I'm doing with my students. And yeah, the lives are saved. They're under that IGTV section. And last year, right before the AP exam, I actually did a bunch of sessions with the Absolute Recap. And so her and I have been talking a lot about that we were going to maybe do some other sessions. We're thinking in the future, near future, we're going to do some on how you can prepare yourself for multiple choice questions and free response questions and how to better apply knowledge, not just content knowledge. So if we have questions about content, be sure to put them down there. I'm going to get back on my going through the different organelles to try to help us make sure that we have our review of Unit 2. Remember, Unit 2 is all on the organelles, and it's on membrane transport. So we talked about the nucleus, we've talked about the rough ER, and we talked about the smooth ER. We also have talked about the ribosomes and how their function is for protein synthesis. So we said that when they were attached to the rough ER, that they're going to allow for membrane proteins. So if we see these ribosomes just hanging out in the cytosol, they're going to make cytosolic proteins. These are proteins that are just kind of free floating in the nucleus, in the cytoplasm, and they can move throughout the cell to do different functions. When you look at cell communication, there's a lot of relay proteins that are responsible for bringing the message to get the type of response that you need within the cell. Let's see. We also have the Golgi bodies. The Golgi bodies have the job of modifying and packaging and sorting the different proteins that were synthesized by the rough ER. And so what's going to happen when we think about how things are secreted or how things move throughout the cell is that the mRNA is going to leave the nucleus, is then going to bind to a ribosome. Now, here's a misconception that a lot of people don't understand, is that all ribosomes start out as free ribosomes. So the mRNA will find a ribosome in the cytoplasm to start for the process of translation, where you read the mRNA to make a protein. And so what's happening is that the ribosome is then going to get a message, and if it gets that message, it then moves to the rough ER to finish translation. And so when we think about the steps, of course, mRNA, ribosome, rough ER, and then when we're in the rough ER, we're going to make that protein. The protein's going to leave the rough ER in the form of a vesicle. Vesicle will then fuse with the Golgi body. The Golgi body will sort it, modify it, do what it needs to do, and then it's going to send it to the plasma membrane to be secreted. It will then go back through another vesicle, then bind to the membrane, and go through a process of exocytosis to release out that protein. Also, this is how different organelles are made. So, for example, your lysosome is made this way. So once we make the hydrolytic enzymes, they're within the vesicle, and that vesicle, since it has these hydrolytic enzymes, it's now considered the lysosome. And the same thing with how we make our peroxisome and a couple other of our organelles. And so what I've mentioned so far, this is all part of the endomembrane system. And so this is a system within the cell that is all connected by membrane. Your nuclear envelope, your ER, your Golgi, your vesicles, your lysosome, your plasma membrane. All of those are connected by membrane. And so what we see is that the mitochondria and the chloroplast are not part of the endomembrane system. We believe that they are prokaryotic organisms that were engulfed long time ago when the eukaryotic cell was being formed. So I'm going to pause and I'll go back to the mitochondria and the chloroplast because it looks like there was a question. What effect does cholesterol have on the membrane? We're going to get back to that question when I talk about the membrane in a second. So we're not there yet. So on the mitochondria and the chloroplast, those are responsible for different chemical processes within the cell. So your mitochondria, its job is for cellular respiration. And so the glucose is going to go through the first step of cellular respiration outside in the cytosol, and it moves into the mitochondria in the form of something called pyruvate. And so when it's in the form of pyruvate, it's then going to go through two parts within the mitochondria for the synthesis of ATP. And I'll go through more on how the mitochondria does the cellular respiration when we do our unit 3 one. But for right now, all we need to understand is that the mitochondria's job is to synthesize ATP. And so it's going to use the chemical energy that's stored in the form of pyruvate to continue to break it down to release that energy and store it in the form of ATP. Those of you who don't know, ATP is adenosine triphosphate. So it's a nucleoside in which you have the nitrogenous base of an adenine. You have the sugar of I believe, ribose. And then you have 3 phosphates attached to it. And so that makes ATP. And when we see ADP, that's adenosine diphosphate. So instead of having 3 phosphates, I now have 2 phosphates. And so the process that happens in the mitochondria is it just adds the 3rd inorganic phosphate that's free floating. So the chloroplast's job is to synthesize glucose. And actually it's synthesizing something called G3P, glyceraldehyde 3-phosphate. Don't worry, you don't have to know that word. And so what's being done is that the solar energy is shining down, which is providing the energy for the cell to be able to make ATP, and then the chloroplast takes that ATP and stores that energy in the form of G3P, which is glyceraldehyde 3-phosphate. And again, we'll talk more about the mitochondria and the chloroplast in our Unit 3 session when we talk fully about photosynthesis and cellular respiration. So big picture, mitochondria is the site of cellular respiration. We synthesize ATP. The chloroplast is the site of photosynthesis, and that is where we're going to synthesize G3P. So other organelles, we have the lysosome that we mentioned before, but I don't think I said what it did. Its job is to break down different damaged organelles or to break down macromolecules. So if the cell, let's say we're looking at a white blood cell, it goes through a process of phagocytosis. Phagocytosis is where we're going to engulf something. So your white blood cell will engulf a bacterial cell that's been marked with an antibody so that it can, again, fight the infection. And so since we brought in, through phagocytosis, we bring in this bacteria, it's now inside of something called a food vacuole. And that food vacuole is going to fuse with the lysosome. The lysosome has hydrolytic enzymes, so basically enzymes that allow for the process of hydrolysis, in which you're going to break a water bond and bond in the water molecule in order to break a bond within a structure. And so we can break down that material. And so hydrolytic enzymes break down this material. So we can break down our prokaryote. We can break down any type of damaged proteins. We can send them there and get rid of them. We can break down damaged organelles. So there's a lot of things that happen within the lysosome to kind of help to reduce things. Now, as I said before, AP exam isn't going to straight say to you, hey, what is the job of the lysosome? They're going to give you a scenario. So one of the big scenarios we always talk about is Tay Sachs. In Tay Sachs, there's an accumulation of lipids in the brain of these individuals. And the reason why is because their lysosome is missing those enzymes that are responsible for breaking down lipids. And so since they can't break down the lipids in those brain cells, there's an accumulation which then has a large impact on their mental capabilities. And if I remember correctly, they do have a short lifespan because of that buildup of that lipid. So it's not, good when these organelles that we have inside of our cells malfunction. So I think I've mentioned the important ones. We said the nucleus. We said the rough ER, the smooth ER. We talked about the Golgi. We talked about the vesicles. We talked about the vacuole. Oh, we didn't mention the central vacuole. So central vacuole is going to be found only in plant cells. And the central vacuole's job is to, of course, store nutrients. But the big job is looking at water. So we'll have a large amount of water that's stored inside the central vacuole. That then kind of pushes on the cell wall a little bit, in the cell membrane on the cell wall, which creates this thing called turgor pressure. And turgor pressure is one of the things that allows a plant to become upright. And as we move into the membrane transport in a second, we'll look at how that is going to impact the tonicity of the plant. And so there's central vacuole has water. Okay, so we said vacuoles. We said food vacuole. This is where we're going to, of course, when we engulf something through phagocytosis. We talked about the lysosome. Peroxisome is also another detoxification organelle. Its job is to detoxify by making something called hydrogen peroxide, and then it breaks down the hydrogen peroxide into water and oxygen. If you've done the catalase lab, traditionally you're using the enzyme catalase that is part of the peroxisome. Its job is to, of course, break down. That's why whenever you get a cut and you put hydrogen peroxide on it, the reason why it's breaking down is because your damaged cells have released out this hydrogen peroxide. Your damaged cells have released out the catalase, and then you put the peroxide on it. Catalase speeds up the reaction of the hydrogen peroxide into water and oxygen. And again, if you have questions, put them in the little question box down there. I'd love to answer questions instead of just freely talking. So we talked about the peroxisome, metabolism. Okay, I think we have all the organelles that we need to mention. Hey, we got some newcomers. Welcome. Make sure you put your questions down there. Okay, so after we had all of those organelles, we also need to talk about the membrane and membrane transport. So first thing we should talk about the way that the membrane is structured. So your plasma membrane is mostly made of two main components. You have the phospholipids. Phospholipids are something from Unit 1 that we learned about, in which you have a phosphate head and you have two fatty acid tails that comes down from it. Okay, fatty acid tails would be hydrophobic, meaning water-fearing, which means they're non-polar. And your hydrophilic head, or your phosphate head, is charged, and so it's hydrophilic and it's water-loving. And so the membrane orients itself automatically so that the phosphate heads face the interior of the cell and the exterior of the cell. And then we have those non-polar fatty acids facing the inside. So if my hand was the circles, our hand is the phosphate heads, what you see is the membrane is going to be set up like this, where it's a bilayer in which you have the phosphate heads on the outside and the non-polar fatty acid tails on the inside. And so based on whether they're saturated fatty acids or unsaturated fatty acids, it's going to allow for polarity within the membrane. And the second component that we have that makes up the membrane, big component, there's other things, but big components would be proteins. So there's proteins in the membrane that are going to allow for, they can have enzymatic functions, they can allow for transport proteins, they can allow for cell recognition, they can allow for cell kind of joining to the extracellular process. Like, so we can attach to the extracellular matrix using something called integrin. So there's a lot of things that can happen because of our membrane. Another part that's in the membrane, it's not like a huge thing, but it's important, is something called cholesterol. Cholesterol is a steroid. And so what it does is it's going to sit in the interior of the membrane at the fatty acid tails. And it's going to allow to be a membrane buffer. So if it gets really, really hot, then it's going to kind of hold the fatty acid tails together to stop them from making big breaks in the membrane that can allow, of course, too much water, too many materials to come in that can cause damage to the cell. And if it gets too cold, it stops the fatty acid tails from compressing, which of course makes the membrane solid. And then we need our membrane to stay fluid to allow materials to pass through. So the components that we need to make sure we know, we have phospholipids and we have proteins. Additional components we have would be, of course, cholesterol. We also have something called a glycoprotein and a glycolipid. Glycoproteins are going to be proteins that are attached to sugars. And so it allows for cellular recognition. And then glycolipids are the exact same thing, in which we have a carbohydrate attached to a lipid. And that's actually how we get the different blood types. So in blood type A, you have the glycolipid A, and then blood type B, you have a glycolipid B. And so those different carbohydrates that stick out from that membrane is what allows us to have A-type blood versus B-type blood. And that allows for, if you had A-type blood and somebody gave you B-type blood, that's how your body knows, oh, this isn't me. And then your immune system starts to, of course, break it down. So the antibodies bind to it, causing coagulation or kind of the clumping of those cells, which is a bad thing, which is why they always test your blood before they give you blood. So nice of them. So let's see. I think we had a question from somebody about the membrane. So let's see. What effect does the cholesterol have on the membrane? So just making sure that we got that down. The cholesterol's job is that it's going to act as a temperature buffer. It ensures that the membrane doesn't get too spacey and doesn't get too compact. Another thing we kind of want to make sure we understand about the bilayer is the reason why it's associating that way is because of the hydrophobic tails are going to go on the inside because they're afraid of the water. They're facing away. So if the interior of your cell was actually hydrophobic, so if we didn't have our interior of our cell made of water, if the interior was hydrophobic, you would only have a single layer because the fact that the outside would be hydrophilic or be water. And so of course my phosphate heads would face the exterior. And then if the interior of my cell was nonpolar, we wouldn't need the second layer because it's nonpolar and hydrophobic likes the nonpolar area. So be sure to think about that when you're working on your practice questions. Also, let's think, what else do we have? Okay, so membrane transport. How does things get across the membrane? So we said before, and again, if you have questions, put them in the bottom. So we said before that the hydrophobic tails are facing the interior of the membrane. So we have the phosphate heads facing the interior of the cell and the exterior of the cell, and then we have these the hydrophobic tails facing the interior, these fatty acids. And so if it's small and nonpolar, it goes through simple diffusion and passes straight through. It is important to know that water can get through. It just doesn't get through very quickly. So there are other things that can pass through, but it's just not going to happen quickly. The small nonpolar items can pass through very quickly, and they get right through, no problem. They're good to go. So like oxygen, carbon dioxide, these are small and nonpolar items, and so they can pass through with no problem at all. We also have like transport proteins to allow for something called facilitated diffusion. Facilitated diffusion is where there's a protein, there's a transport protein. There's two different types. You have channel proteins and you have carrier proteins. So channel proteins are going to allow for a hydrophilic tunnel, if you may, that passes through. Now, these are very specific. When I graded the exam this past summer, there was a question that I graded about these channels, and it asked why only calcium went through this channel that I was looking at. And it had to do with the fact that actually, I'm not allowed to talk about this. Sorry. I signed a DNR, no, whatever. I can't disclose what was on the exam. Sorry. Anyways, so channel proteins, you need to make sure that you understand that the channel proteins are specific for the ion that travels through them. And so if we have, of course, a calcium ion channel or a sodium ion channel or a potassium ion channel, then we're looking at that the ion is going to be specific for that channel protein. It's going to bind to it. It'll have the right polarity. It'll have everything it needs. It'll be the right size to allow for that ion to pass through. It binds to the active site perfectly. We also have the carrier proteins. Carrier proteins' job is that they're going to bind and then they change shape, which will allow for carriage across the membrane. So it transports it across. And so the absolute recap has already said the aquaporins. Aquaporins are a specific type of channel protein that will allow for the water molecules to pass through the membrane. And it looks like Hector down there has told me the sodium potassium pump, which is an example of a carrier protein. But he's going more for active transport instead of passive transport. And so active transport is going to be when we're going to move against our concentration gradient. So the previous two examples we talked about with facilitated transport and simple diffusion, those were looking at passive, in which you're moving from a high concentration to a low concentration. And then in active transport, we go from a low concentration to a high concentration with the input of ATP in energy. And so we have okay, I talked about that. Bulk transport, we already mentioned a couple of these words, but just to make sure we have them. Exocytosis is when we're going to have a vesicle fused with the membrane, opening up, releasing out the materials. And so we can allow materials to exit our cell. Endocytosis is when I'm going to bring things into my cell. So I always tell my students exo, EX, is going to exit the cell. Endo, EN, is going to enter the cell. And so there's three types of endocytosis. You have phagocytosis, which is going to be cellular eating. It's going to allow for these little pseudopods, fake feet, that will extend out and surround the material to bring it into the cell. We also have something called pinocytosis. Pinocytosis is cellular drinking. The job is that it's kind of gulping the extracellular fluid, and it brings in a couple of the solutes at the same time. And the last one we have is receptor-mediated endocytosis. So one of the proteins that we have on the membrane can be a receptor. And so if a signaling molecule called a ligand binds to that receptor, it can cause the invagination of the cell. So it kind of takes the plasma membrane and pushes it in and makes a little pit, so that they can bring in specific materials due to that ligand binding. So those are our three different types of bulk transport. So let's think about osmosis. Osmosis is where we are going to have the transport of water across the membrane. And it's due to the aquaporins, as the absolute recap already told us. And that's a channel protein. And so materials are going to move from from high to low. But the problem is, and this is a big misconception with students. Students have big issues with this, okay? When they tell you concentrations, they're telling you the concentration of the solute, not the solvent, which would be the water. Osmosis is looking at the water movement, that water transport. So those numbers do not actually tell you how much water there is, okay? So the lower solute concentration means there's a higher water or free water concentration. If you have a higher solute concentration, it means there's a lower free water concentration. And so when you're looking at these, you're always seeing this moving from a low concentration to a high concentration because you're looking at the solutes. So water would move from a 0.1 molar to a 2 molar solution because it's trying to bring the water over to kind of dilute out that solute. And so there's three different tonicities we could have. We have isotonic, which means that the solution on the inside of the cell and the outside of the cell are going to be the same. They have the same concentration, the same amount of free water. So there's going to be equal movement of water into the cell and out of the cell. And then we also are going to find that there's hypertonic and hypotonic. And so I always tell my students that these are in comparison to each other. If the inside of the cell is hypotonic, the outside of the cell is going to be hypertonic. If the outside of the cell is hypotonic, then the inside of the cell is hypertonic. So they're in comparison to each other. So the questions that they give you on the exam is going to tell you this solution is hypotonic. Okay, and you're going to have to know, okay, well, if that one's hypotonic, this other solution is going to be hypertonic. Water is always going to move from a hypotonic area to a hypertonic area. Let me say that again because it's very important. Water is always going to move from a hypotonic environment to a hypertonic environment. So water is going to move whichever direction we need to move to go from hypo to hyper. So let's think about animal cells for a second. Animal cells have no cell wall. And so it's like a water balloon. You fill it up with water, sooner or later it's going to burst. So if we take an animal cell and we put it in a hypotonic environment, so because the animal cell is in a hypotonic, the cell itself is hypertonic. Water is going to move into that cell, which is going to cause the cell to burst or lyse, which is not good, okay? There was a free response question one that I read 3 years ago, and I can talk about this one because this one was released, in which they were looking at cell communication, and it was talking about that a bacteria cell had invaded these cells. And so the way that the cell kind of protected itself was it added this pore called gastrodermin. And the gastrodermin was going to basically make water come into the cell. And so as the water ran into the cell, it caused the cell to lyse, which would damage the host. So the bacteria wouldn't be able to have a host anymore because it basically killed the cell. So that's how it protected itself. So if we had that same animal cell, and instead we put it in a hypertonic environment, that's going to mean that the outside is hypertonic, so the inside of the cell is hypotonic. So the water is going to leave the cell, which is going to cause our cell to shrivel. Okay, it's called crenate, but basically just means it shrivels up. And then if you have an isotonic environment, same amount of water, we're good to go. And that's actually normal for an animal cell. But the difference is in our plant cell. And we take our plant cell and we put it in a hypotonic environment. Okay, water rushes in, but because of the cell wall, it doesn't burst. And this is what I was talking about with the central vacuole earlier. The water goes into the central vacuole, and the central vacuole kind of pushes on the plasma membrane, and the plasma membrane pushes on the cell wall. And so because of that pressure, it helps to give rigidity to our plant cells. So this is actually normal for plant cells. Plant cells should be in a hypotonic environment, okay? Be very careful about the ions. We don't really want the ions to leave the plant, but we do need to be in a hypotonic environment so they can keep its water, and water constantly is rushing in, so that it can be rigid. If we put it in an isotonic environment, it's going to be something called flaccid, so it's just kind of limp. Ooh, my light just turned off. And then the third thing that we could have is where it's if it's in a hypertonic environment, we're going to have plasmolysis, in which the plasma membrane is going to pull away from the cell wall, which is going to be even worse for the plant. So, there any other questions? I did a quick review of our organelles. I've done a quick review of membrane transport. I'm going to give y'all, if there's any questions you have, make sure you're putting it down there. If not, then I'm going to end it just like I did last time at 30 minutes because I only want to do this for what you guys need. And I'm not going to waste your time if you guys don't have questions. So if you have a question, be sure to put it down there. I'm going to kill a little bit of time while I wait for anybody who does have questions. Make sure you do follow. There's 2 other accounts in case you're taking other AP exams that, you might need some help with. So the Absolute Recap does biology, chemistry, U.S. government, music theory, and physics. And so the Absolute Recap is actually a podcast. It's always less than 10 minutes to kind of help out you to have this quick little refresher on content or learning of content. And then Marco Learning actually does U.S. history, AP Lang. They do college prep like SAT and ACT prep. And then they do AP language. They may do other ones, but those were the ones I was able to quickly find when I was kind of looking at what they did. So if you are taking any other APs, make sure you do follow Absolute Recap and Marco. They're both amazing. So there's been no other questions submitted down there. So I hope you guys have a good weekend. And bye, y'all.